A point mutation is basically a mutation which involves the switching of one base for another. For example, an adenine would switch with a thymine, or cytosine, or guanine, or any number of those combinations. A transition point mutation is a point mutation which involves the switching of a pyrimidine to a pyrimidine, like thymine to cytosine, or a purine to purine, like guanine to adenine. A transversion point mutation is a point mutation which involves the switching of a pyrimidine to a purine, like guanine to thymine, or a pyrimidine to a purine, like cytosine to adenine. A missense mutation is a single point mutation in which a single nucleotide change results in a codon which codes for a different amino acid. In this case, cytosine in the coding strand changes to adenine, which results in guanine changing to thymine in the template strand, and then cytosine changing to adenine in the mRNA. This causes the codon to code for lysine instead of asparagine, which results in a change in the protein function. This is also an example of a transversion point mutation. A silent mutation is a single point mutation in which a single nucleotide change results in a codon which codes for the same amino acid. The amino acid in this mutation remains the same. In this case, thymine in the coding strand changes to cytosine, which results in adenine changing to guanine in the template strand, and then uracil changing to cytosine in the mRNA. However, the codon still codes for alanine and there is no change in the protein function. This is also an example of a transition mutation. A nonsense mutation is a single point mutation in which a single nucleotide change results in a codon which stops translation. In this case, cytosine in the coding strand changes to adenine, which results in guanine changing to thymine in the template strand, and then cytosine changing to adenine in the mRNA. In this case, the resulting codon stops translation and shortens the original protein, which changes its function. This is also an example of a transversion mutation. A readthrough mutation is a single point mutation in which a single nucleotide change results in changing a codon that would stop translation to a codon which codes for an amino acid. In this case, adenine in the coding strand changes to guanine, which results in thymine changing to cytosine in the template strand, and then adenine changing to guanine in the mRNA. This results in tryptophan being formed instead of translation stopping. Because of this, the resulting protein's function will be different than the original. This is because its length is longer. This is also an example of a transition mutation.